started. Thank you all for joining us today. This is the final webinar for GMAC Region 3 this year. And today our speaker is Dr. Angela Wandinger-Ness. Dr. Wandinger-Ness earned her PhD in biochemistry from UCLA. She's been on faculty in the Department of Pathology at the University of New Mexico since 1998 and has been continuously funded as a PI since 1992. Dr. Wandinger-Ness currently serves as a full member in the Cellular and Molecular Oncology program at the University of New Mexico. And in addition, she's also the Associate Director of Education, Training, and Mentoring for the UNM Comprehensive Cancer Center and the leader of the Cancer Research Training and Education Coordination Corps. She has personally trained 73 students and fellows and served as an advisor and mentor for 131 students, 46 fellows, and 11 junior faculty. We're excited to hear from her today, and I'm going to turn it over to you, Dr. Wandinger-Ness. Thank you.
Sorry. Thank you very much, Shoshana, for that kind introduction. And it's really a pleasure to be here with you today. I'm going to try to toggle between my PowerPoint and a few active audience participation activities. So I hope you'll bear with me as we do that. Let's see, I gotta find the navigation. So I'd like to start off with the question, since the title of my talk you saw before is Women in Research and Leadership, Challenges and Successes. And I'd like to start off by asking the audience as to what is your driver in science or research. And to do that, let's go, I'm going to activate this poll, and you should be able to go to my link. Let me find that, sorry, which is, if you go to pollev.com and then type in Angela Ness 677, you should be able to enter your answers there.
Can you repeat the URL?
Yes, here. It's, if you just go on your browser to pollev.com and then Angela Ness 677. You should be able to see it at the bottom of this slide here too. I'm gonna paste it up on this slide too. All right, we see an answer. Yay. All right, it works. So thank you so much for participating. Here we have a few more. So improving health is a top priority among this group. All right, that's the holiday spirit. All right, so we have lovely answers here, and this helps me to know who's in the audience. I'm going to let you know that I'm a basic scientist, so I have come into health and healthcare research, clinically focused research through my interest in basic research, and I'll explain a little bit more about that today. So I'm going to return back to my, sorry. I need to go here. Did I stop sharing? I did. Bear with me. share. All right. Okay, so thanks for those nice answers. My path to discovery and an innovation includes three main pillars, and they are my passion for life and research in general, my excitement about collaborating with others, and my commitment to diversity. And you'll see those threads through the things that I'll present to you today. Early on, I was an undergraduate at the University of Massachusetts in Amherst, and I had the pleasure and opportunity to work with Bruce Jacobson, shown here on the right. And his lab was studying the slime mold Dictyostelium, which in the wild ingests bacteria, and it is very migratory, but we were interested in using it as a model for understanding the changes that happen as this slime mold interacts with its environment and how it remodels the cells, its own cell surface, in detecting either food or moving towards food and things like that. And at the same time, there was research ongoing about cancer cells, and cancer cells had markedly different plasma membrane or surface compositions compared to the normal cells in the body. And these two things were connected for me because we were looking in this slime mold model at the cell membrane, but yet there was a human application in that cancer cells changed their surface composition and they became more fluid or more liquid, which allowed them to also become more migratory. And this theme, I'll come back to the cancer theme in a little while. So after I graduated with my bachelor's of science in biochemistry, I was really tired of courses where people told me what I needed to learn. I was really interested in pursuing the things I wanted to do, and that's the passion, really. So I moved to Maryland and worked at the University of Maryland, Baltimore County campus, and that was a brand new campus at that time. It's gained a lot of recognition because of Freeman Hrabowski, who was the president there for quite some time, and most recently there's a Diversity, Equity, and Inclusion Award that's been named in his honor. I worked with Donald Creighton at UMBC, and he took me into his lab really on a promise, right? That I would work hard as a post-baccalaureate technician in his lab. And what we studied was a protein or an enzyme that's derived from papaya, and it's called papain. And this work was very detail-oriented, and we were trying to figure out how the enzyme worked. And it led to my first publication in Feb's Letters. So this was a thrill for me, and it reaffirmed my interest in pursuing a higher degree in science. And Donald Creighton had come from the University of California, Los Angeles. And since I spent all of my childhood and my education on the East Coast to that date, I wanted to go to California and see what opportunities lay there. And so off I went. And at this point in time, as a first-year graduate student, I met a boy, and I'm still married to that boy 37 years later, and he has been my steadfast companion, both in science and in life. And so at UCLA, I pursued a PhD in biochemistry in the laboratory of Richard Weiss, where I was studying protein biochemistry, genetics, and metabolism. Arginine biosynthesis or arginine metabolism in cancer has gained a lot of recognition recently. So it's interesting how things come around. And I think that's an important theme of my talk, how you can build on skills and you reutilize them in the future. Although I haven't returned to arginine metabolism, I did work out how arginine is made, the first steps from glutamate in the cytoplasm, and then converted into the final product out here in the cytoplasm. So it goes from the cytoplasm to the mitochondria and to the cytoplasm again. So this is very basic research. It was conducted in a different kind of mold, in a Neurospora crassa, a bread mold, and it really used my skills that I had gained as post-baccalaureate student in terms of doing protein purification, biochemistry, and it moved me a little bit into cell compartmentation, and I'll come back to that. So from there, my path continued and took me from the US all the way to Germany. And in Germany, I worked at the European Molecular Biology Lab with Kai Siemens, who was a group leader there. And it seemed like it wouldn't be such a big change, but it was a huge change in what I knew about science. I began to integrate biochemistry, but with a whole new field, which is cell biology. And I learned about membrane trafficking, which is the process whereby proteins or molecules are moved within a eukaryotic cell to various destinations. They can move to different membrane surfaces on this, again, recognizing the importance of membranes of the cell. And then also materials could be, this is how cells communicate with external environment. They can absorb nutrients and respond to signals in a complex system, a mammal or a human. They're receiving signals from the bloodstreams and responding to those. And so I was interested both in how molecules were moving from inside the cell to the surface or being secreted, and how they were being taken up and how cells were responding and changing in response to that. And some of the important cells that really are pivotal everywhere in our body are epithelial cells. They coat the surface of our body, our skin cells, but also all of our organs have epithelial cells. And so these cells are special in that they're polarized. So they have two different surfaces, and we were interested in how that is set up and maintained. And in cancer cells, one of the things that happens, you often lose cell polarity. I was studying all of this in the context of kidney cells, and I'll show you why that's important in a minute. And then I was also looking at enzymes that are important controllers of these processes that I've been describing to you. So from Germany, then, I took my first faculty appointment at Northwestern University in Evanston, Illinois. And here I was mentored by senior colleagues, and I was able to get a National Science Foundation Career Award. And this was really important to me because it combined research. I had to have a research plan, but also a plan for teaching and education. And ever since I was at UCLA, I had an interest in education and teaching and mentoring. And so this NSF Career Award really promoted that interest of mine. Additionally, I received a grant from the National Institutes of Health, an R01 grant, and that grant focused on, together with a colleague whom I met at the European Molecular Biology Lab, Dr. Robert Bacalhau, that grant was focused on a human disease, an inherited disease called autosomal dominant polycystic kidney disease. So now this project could bring together my basic understanding about cells and the biology of kidney cells and put it into a whole organ context, as well as in the context of human health and disease. And so what happens in this disease is that you can inherit a mutant copy of the gene that causes this, of one of two genes that causes this disease. And when that happens, this is autosomal dominant, which means that when you inherit one mutant allele from a parent, then it will result in a manifestation of the disease. Although the disease severity can vary depending on the other genes that or alleles that a person may have. And so what happens in the disease is that the kidney has this functional unit that is responsible for having the blood come in and get filtered here. And then the urine gets concentrated as it flows through this nephron. So you can imagine this like a tube, like a garden hose, and lining that garden hose are the epithelial cells that I was talking about a moment ago. And so those epithelial cells are either secreting or getting rid of the waste into the garden hose, and then absorbing the things that should be returned to the blood out of that hose. And ultimately, this structure goes to the bladder, and then the urine gets excreted. And what happens in the disease, as you can see in this picture, is that this whole nephron gets these cystic structures all covering it, and that causes this compression of the normal functioning of these units in the kidney and ultimately leads to kidney failure. And so our contributions here were to take biochemistry, cell biology, and our understanding of GTPases and how proteins move in the cell to understand something about this disease. But in the meantime, I also moved onwards to New Mexico. And as you'll see here, there were two wonderful women who became my champions when I moved here. Mary Lipscomb, who was a chair of pathology at that time, and Cheryl Willman, who is the Comprehensive Cancer Center director, or it wasn't comprehensive at the time, but she was the champion for building a comprehensive cancer center here. And so this is the first time that I had women as my role models. So I carried on then my project that I had been funded at Northwestern at the University of New Mexico. And I had a team of investigators. Here you see they're all women. Elsa Romero was she became my first tech and only tech at the University of New Mexico. Tamara Reutbach was a PhD scientist. Heather Ward, also a PhD scientist. Stephanie German was a graduate student in my lab. Emelina Silverberg was an MD, and she really motivated me to isolate the cells directly from human disease tissues so that we could better understand the disease processes. And so I've really been benefited in my career through these interactions with trainees and staff who've helped to move projects forward. As I mentioned already, if you have polycystic kidney disease, if you have either one of two mutant genes, and these genes encode two proteins that sit at the membrane of the cell, where they're important in sensing and signaling to the cell. And when these proteins are mutant, you get this massive rearrangement of the kidney, as I showed you already. So it seems like a small thing to have one mutation to cause so much devastation. So how might it occur? What we discovered is that some of the proteins that are mutant interact with key components that allow the cells to connect to one another and shake hands and keep that normal kidney architecture going on. And also, these proteins are important in this antenna-like structure that's on the apex of the cell and faces towards the center of the garden hose, and it recognizes the fluid flow of the urine going out. And so again, these proteins are important in two ways in sensing in the cell. And when they are mutant, then these two proteins, the cell-to-cell contact and cell-to-substrate contact, as well as this response to fluid flow, is disrupted, and that may be important in the disease process. And as you'll see, we use different tools to determine what is going on. And here you can see normal human kidney cells that we've we've plated them in tissue culture, so they're growing here. Here's one cell next to another, and here are those cilia or those antennae that I mentioned. In the normal cell, they have those antennae and they have the normal polycystin protein present. And in the polycystic kidney disease cells, the antennae are there, but the polycystin protein is absent from this antenna. So it's not the loss of the antenna, but it's the loss of that mutant protein from the antenna that may be important in the disease. So let's return back to these enzymes that are important for mobilizing proteins through the cell, either inward or outward. And here again, I had a whole wonderful group of postdoctoral fellows, undergraduates, and graduate students who worked with me on various aspects of this project over the years. And just to say, they've gone on to their own successful careers. Sam Schwartz here is a postdoctoral fellow. She got her PhD at UNM after being an undergraduate in my lab, taking a post-baccalaureate break like I did. Jacob Agola is an engineer at Intel. He was a graduate student in the lab. Mateos Tessima was a postdoc, and he's a doctor of veterinary medicine. He's now at Loveless Respiratory Research Institute here in Albuquerque. Sanchita Mukherjee was a postdoctoral fellow and is in the private scientific sector and has moved between doing bench work to editing and so forth. And Shomik Bazare is currently at Novartis as a science officer. So science is never a straight path in my experience. We talk about leaky pipelines or pipelines, period, and then leaky pipelines. But actually, I find that it's more of a pathway and there are twists and turns along the way. And it was 4 years into my appointment at UNM. And I heard the word cholangio and said, what is that? And it turns out it's cholangiocarcinoma. It's a deadly disease of the liver, and it's a tumor that affects the bile ducts. My father was diagnosed with this in 2001 and passed away from this in 2002. So this really reaffirmed my commitment to turn my research even more strongly towards improving human health, and particularly trying to identify opportunities to make a difference for cancer. And it prompted me to build new collaborations with my colleague, Lori Hudson, who had also come from Northwestern, except that she was at the downtown campus and I was on the undergraduate campus. So we didn't really interact much during our time at Northwestern. But I approached Lori and said, can we do something together on ovarian cancer? Because this was and still is a devastating disease with a dismal outcome. And so we started working together and also bringing in the idea of using these enzymes that I mentioned that are important for moving proteins to the cell surface and that can change a cell membrane or change the signals that cells receive. And could we use those enzymes as targets for developing better treatments for cancer? And I worked also together over many years with Dr. Larry Sklar, who's a colleague in the Department of Pathology, and Lori Hudson is a faculty member in the College of Pharmacy in Pharmaceutical Sciences. And so some of the work that we did to try and build therapies was to use a high throughput screening strategy whereby we would try to identify drugs or small molecules that could inhibit the activity of GTPases. And we, had a number of great funding from the NIH. And our goal was really to trigger the switching of GTPases from an on state in which they're they may be continuously active and may be driving the cell forward in a metastatic process in cancer, for example, and how could we turn off that switch? So that was our goal. And this kind of work takes a team. So here you see Dr. Hudson, myself in some early days where we had received money from the Department of Defense and to do some clinical studies. And we did those together with Dr. Carolyn Muller, who's a gynecologic oncologist here at the UNM Comprehensive Cancer Center. And so we moved these studies from a very cellular cell biology side, mechanistic side, together with Lori, all the way to mice, testing the drugs in animals, and then on to doing a clinical study with Dr. Muller. So what we achieved through this work, which has really gone on for almost 2 decades now, is to identify, to achieve our goal of finding molecules, these small molecules, drug-like molecules that can block or inhibit GTPases. We were also able to test some molecules in humans with Dr. Muller, and these have potential applications. We tested them in ovarian cancer, but may also have applications in breast, colon, and lung and prostate cancer. And with Dr. Linda Cook, who's a member of the Cancer Center for many years and is now at the University of Colorado, we did a small pilot study, a retrospective study, as well as a prospective study with Dr. Muller, and showed that there seemed to be a survival benefit when patients received ketorolac, which is a drug normally given for pain relief, but it comes as a mix of 2 different chemicals. One, you can think of this as your right and your left hand, and one of the 2 chemicals, they're otherwise identical. The one that is called S-ketorolac is the pain relieving medication, and the one is R-ketorolac is the one that inhibits these GTPase enzymes. And so that's how we were able to move this into the clinic, by giving the drug for its clinically indicated purpose, which is pain relief. But at the same time, we could see and study what happens to the tumor cells in response to the presence of this GTPase inhibitor. So I'd like to pause just a moment and see if anyone would like to ask any questions about the research side of the house or anything that I've mentioned so far. Chat. There's some questions in the chat. I can't see them. Oh, okay. No, not really. Anyone? Okay. Then I will carry on. So I'm going to turn now towards the leadership component and talk a little bit about my path in leadership, but also to talk about some of the challenges and some of the resources that are available in terms of leadership, and without being comprehensive necessarily here. Okay, so for myself, I have to say that I was coaxed into institutional leadership roles by the 2 wonderful women who are leaders in their own right that I mentioned already, Dr. Cheryl Wellman and Dr. Mary Lipscomb. And so one of the first roles that I was asked to take on, and it was just a few years after I got here, I guess 7 years after I got to the University of New Mexico, Dr. Lipscomb asked me to take on the role of the Assistant Dean of Graduate Studies at the School of Medicine. And this graduate program, the Biomedical Sciences graduate program, is one that serves the whole health sciences campus, College of Pharmacy, faculty, and others across the campus educate students from this program. So I was really hesitant to take on this role. I felt like I was premature, I wasn't ready, and Mary just kept at me and convinced me to do this. So here's one point I have learned in my life and been given this advice, and that is if you're not sure about a leadership role, then you can certainly say, sure, I will try that. I will do my best, and I will do it for this amount of time. And this was advice actually given to me by Dr. Lori Hudson. And she said, you don't have to do this with no end date. Why don't you do it for 3 years? And so that's exactly what I did. And this was a much more bite-sized chunk because I could say, these are the goals that I would like to accomplish during this period of time. And one of those was to revise the biomedical sciences graduate curriculum. It was up for academic program review, which is a big deal. It's organized by the University of New Mexico, but it entails recruiting outside reviewers, and they come in and they review everything about the program. They speak with the students, they speak with the faculty, they review the curricula, they judge the satisfaction with the program and all of that. And we got quite nice and positive reviews on this. And so I was pleased with that we had done a curriculum revision and that was well received. And the students were feeling that they were getting value in the education that we were providing them here. And I thought that was, for me, that was an important outcome, let's say. Additionally, because of my passion in teaching and education at the Health Sciences Center, we really didn't have much of a program to help graduate students prepare themselves for an academic career where they might have to do some teaching, or for that matter, even to give oral presentations. And so I implemented with my colleague this university science teaching certificate, which requires lots of formal documents and platforming and testing courses that you want to be part of the certificate, but it is available for graduate students to get this transcripted on their degree. So in addition to earning their PhD in biomedical sciences, they get a transcripted certificate on their diploma. So after, or even actually concurrent, and you can see why I was a little reluctant to be the dean here, I established a fluorescence microscopy shared resource at the University of New Mexico Health Sciences Center. And this was because I showed you some pretty images before that are taken on a fluorescence microscope and are really essential to a cell biologist to see what is happening within the cell and the subcellular structures. So when I arrived at UNM, there was no what is called a confocal laser scanning microscope. It is a light microscope, but it gives you very high resolution because it uses a laser, and so it can scan one line at a time, and you can imagine it going down one little bit at a time through the entire section of a cell, thickness of a cell, and can give you information throughout that cell. So I set this up and was the head of that resource for 16 years until I passed it off to one of my colleagues. Now, here's a situation where I hadn't actually set a deadline, but I did plan my exit strategy with my colleague. And that was important because this shared resource became part of the UNM Comprehensive Cancer Center and a NCI awarded P30 grant in support of that center. Again, returning to my interest in education and training, I applied as the principal investigator for an NIH K-12 grant in support of diversity in science research for postdoctoral fellows. And I've been the program leader of this since 2009 and will hopefully renew it again in 2023 for the third cycle. And currently, I still work at the UNM Comprehensive Cancer Center as Associate Director for Education, Training, and Mentoring. I've been in this role since 2013 and formally as funded or supported by this NCI P30 grant since 2020. And in that role, I'm the Associate Director for Cancer Research, Training, and Education, which is a core function, which means that we host activities for trainees from high school all the way up to the junior faculty development levels. And finally, most recently, I was asked a couple of years ago to become a member of the University of New Mexico Health Sciences Inclusive Excellence Council, where I serve as an executive officer. And one of the most important things we're doing right now is a institutional climate survey to assess where we can improve our efforts in the context of gender diversity, of diversity at all levels, and also equity and inclusion for trainees, staff, and faculty members. So in addition to the institutional leadership roles that I've mentioned, I have been an active member of scientific societies, which these offer leadership opportunities that are not as high stake. For example, I served on the Women in Cell Biology Committee. And what was nice about this was a group of colleagues from across the country. And we were recognizing women who had made important contributions to science. And I also served on the American Society of Investigative Pathology. This was at Dr. Lipscomb's urging that I be a member of this society as a member of the pathology department, and it was great because I served on the education committee, as I mentioned. That's one of my passions. And currently I'm on the great group for the American Association of Medical Colleges, and particularly focused on how to improve postdoctoral training and education, where there are some real gaps because postdocs tend to be recruited to individual laboratories and often don't get the mentoring and training that's, for example, graduate students might have in a more structured program. So the scientific societies is a nice way to take on leadership roles, in my opinion, that where you can work with colleagues and learn lots in the process. I've also served on many grant review panels, and this is also very both educational and a way to pay it forward. If you've been the recipient of a grant, you can help to provide constructive comments to colleagues so that they can continue to improve their work, and also to continue their work through the funding that is afforded by these agencies. In addition to federal agencies, I have served on review panels for private foundations, the Polycystic Kidney Disease Foundation, and most recently for these privates that are awarding recognizing achievement both in science and diversity, equity, and inclusion. One of them is the Freeman Hrabowski Award. As I mentioned, he was the president at UMBC for quite a while, and this is being sponsored through the Howard Hughes Medical Institute, as well as the Chan Zuckerberg Awards that are also in the same vein supporting diversity and scientific excellence. And finally, I've served on award selection committees like the Presidential Award for Engineering, Science, Mathematics, and Engineering Mentorship, as well as the American Association for the Advancement of Science, Lifetime Mentor Award committees. So that has also been a great pleasure and wonderful to interact with colleagues around the country and get to meet others like-minded colleagues. So let's go back now to ask the audience this question. What are the magic powers that you would bring or you do bring to a leadership role? And so if I can go back here, deactivate this guy, and we'll go on to the next question. And so just like before, we're going to answer this poll now. your magic powers. I think it's active, right? There we go. Lovely.
Angela, if you're
Yes.
screen sharing, we can't see right now, or
You
it's not
can't see it?
No.
Okay, let me see what happened with the screen share. Oh, maybe did I drop out of it? Oh, I did. So let's go back to the desktop. Okay, now can you see it now?
Yes, thank you.
All right. Isn't this fun? I love this. Those are great answers. All right. Is there some other? Yeah, so we got some great answers there. And I wanted to ask you all these questions before I show you some of the traits that were given as top 10 essential traits by the Center for Creative Leadership. And this is not intended to be the end all and be all. I think it just shows some of the things that you actually also pointed to in your answers. And for me, as a leader, I try to recognize my limits. I mentioned that already. Facilitate connections. I really love connecting. If I don't know the answer, I like connecting people with one another that may be able to find the answers together. I like to think outside the box and to find solutions. So those have been things that have been my guideposts. I also thought it would be important to provide some resources for leadership that might be of interest to the group. The advanced program was funded for almost 20 years by the National Science Foundation and the University of New Mexico, along with over 177 institutions across the country in 47 states have had these advanced program awards, and they were mostly focused on women, advancement of women in academic science as well as in engineering, where it was recognized quite some time ago, back in 2001, that there was a real need to promote women in engineering and some of the physical science careers, as well as in STEM careers in general. And although the advanced awards have sunsetted, this has some wonderful resources are still available through advance, and I gave the link here. Nonetheless, even with all these programs, and we're 20 years further down the road, we recognize this is written just a few years ago, that there still is a need to improve the participation of underrepresented individuals in the STEM careers, in addition to women, and that people refer to this as a leaky pipeline. But actually, I'll show you a different metaphor in a second. And what we can see is that even though there are large numbers of women who start off in getting their degrees in the STEM fields, many of them end up leaving careers in those areas for a variety of reasons. And so I think it's important for us as a community to keep this in mind and to encourage those young people who are coming along and helping mentor them and helping them to persevere and connecting them with peers and helping them to build their networks, because I think that's how we can stem the tide, let's say, and also that they're allowed to bring their whole person to the table. This is something that I had not heard of before, but one of the challenges that women face when they're asked to take on leadership roles is that often they're given impossible tasks. They may be promoted to a leadership position, for example, during a crisis. And this could be a woman, this could be an underrepresented minority, could be any number of people who get put into an unfortunate position as a leader and have an impossible task of fixing a broken culture. And so then they leave, and they call that the glass cliff instead of the glass ceiling, which many of us have heard about. So I think being aware of situations like that as senior faculty and making sure that individuals do not get placed in such unfortunate situations. So we mentioned the leaky pipelines. And what can we do to support women's leadership in STEM? I mean, I think that it's important to reframe what we talk about as the face of technology and women's role in that technology, in science and in medicine, that we also help to scale up interventions that occur early in careers. I think most of us could probably agree that we wouldn't be here if we didn't have a champion at some point along the way that helped us go on. But we have to recognize that for every champion, there might be 10 naysayers along the way, and each person's willingness to persevere in the face of naysayers or in the face of imposter syndrome and all these other things that we've all heard about may be different. And so how can we be people who intervene and give that hand out, reach out to that person right when they need it, and to help them to persevere? And then finally, I think we're working on this, but it's at various stages at different institutions, and that is to reform the recruitment processes and the workplace culture. We hear a lot about it, and we all do lots of training around it, but I think this is something that we have to keep front and center in our minds as we go along. And I mentioned the pipeline, the leaky pipeline. I really prefer this articulation by Annabong Hannah Branch, who was at the University of Massachusetts, my alma mater, and is now Senior Vice President for Equity at Rutgers University. She and a lot of other colleagues wrote this book about pathways, potholes, and the persistence of women, and this can be others who are underrepresented in science, and how do we reconsider this pipeline? And that's why this resonates with me, because as I showed you, my path has not been straight, and it hasn't had just one off-ramp or one on-ramp. It's all about taking turns and changing directions when it makes sense for you. And so that, I think that keeping that in mind as you navigate your career, just because you're making a change, celebrate it, move forward with it. And even if it's not where you had hoped it to be, then just make another decision, a positive decision to do something else. And don't beat yourself up saying, I coulda, woulda, shoulda done that instead of this. No, it's really about taking the chance, taking a turn on something that you think looks great at any given time, and then taking another sidestep if needed. So I feel that success is not necessarily a straight and narrow road. And maybe it's more so for women or others, but I think it's important to bear in mind that there are positive benefits of that. I had mentioned the advanced program. Here are some resources that are available through the advanced program. And then there's also the ARC network that has been established. And there's a nice video that I was hoping I might be able to play here for you. This is very short.
All she carried was only a welcome to the podcast. So excited to be a transformative approach to STEM equity in higher education. What we will do is achieve STEM equity by 2025 through an intersectional, intentional, and inclusive lens. We want to be intentional about our approach. That means not just focusing on what's been done in the past, but making sure we look forward to the future and making sure you're included every step along the way. We also want to make sure that we have an inclusive lens, meaning that we don't just ask you to come and join our community, but we actually engage you in the community. We will be focusing on intersectionality. So not just looking at traditionally excluded identities, but looking at the whole person. We want to make sure that if you're a woman of color or a person with a disability, that you feel included at the table. We encourage researchers and practitioners to join the network to build a community. We also research critical topics in STEM equity to ensure their research agenda moves forward. And the ARC network curates. We'll focus on curating resources to make sure that they are available and accessible to a wide variety of audiences dedicated to STEM equity. Our virtual visiting scholars will be responsible for conducting an analysis on the great research and how that work can inform interventions and policy changes. We'll have an exclusive portal for our ARC network members to join. You'll be able to participate in webinars, live chats. You'll have an opportunity to message other members of the community. I want you to trust that the work that you've done will be valued and appreciated and shared with respect. I want you to remember to be authentic. The only way we will achieve STEM equity is if we remain our true selves, dedicated to making sure that higher education is a place for all.
So I hope that may resonate with you. It did with me. And so that's why I wanted to share that with you. So we've talked a little bit about the leadership pathways and some of the challenges for underrepresented individuals and women to leadership. There may be barriers in the early career stages where, it's often that people are nominated for leadership roles, and the first people to come to mind may not be those who might be exceptional at it. And so those could be barriers for women and others. Often women feel they hold themselves to higher expectations, or others hold them to higher expectations. I mentioned about being brought in for crises. There are often less rewards given for women in leadership, and sometimes the type of research that we do, for example, it was given as a community-based research, may not be recognized at the same level. And it doesn't make sense to me personally, but apparently the types of research may be differentially valued. And how do we make that be viewed differently? I think by being ambassadors is one way that being champions of the work that we do. We need to advocate for our work and make it public and go out there and make other people believers in it. In terms of challenges in leadership roles, there are oftentimes these, we see it at the University of New Mexico, interim appointments in leadership, and then sometimes those get transitioned to a permanent position, but often women and others may go through multiple appointments in leadership before being offered a permanent role in leadership. We may be asked to do more to prove ourselves, and you may have to move out. So change universities, change jobs in order to get that leadership position. And so sometimes you need to think about the willingness to get a promotion or to, I moved here. That was also a promotion for me. So anyway, sometimes we have to do that. It's not always something we want to do. And then I mentioned my leadership roles. They were somewhat more accidental than strategic at times. And so how can you be strategic? Think about how that might make that part of your plan. And then how do you make a difference as a leader? I think we're very close to time. I have just a few more points. There are workload inequities. For example, the work that women and others who are passionate about diversity, equity, inclusion do to mentor and to teach is often not rewarded as highly as our research scholarly works. And so it's important to try and work proactively to address some of those inequities. And one thing that can be done and is articulated here is to develop systems that make it transparent how workloads are divided within a department, ensure that service work such as mentoring and diversity work is rewarded and recognized in systems of evaluation. And then also to prevent the discrimination. So I like this analogy of, instead of there being a glass ceiling, sometimes women are stuck to the floor. And how do we make that floor less sticky so that they can move up and break through that glass ceiling if it exists? So what we can do is to identify role models, allies, and mentors. We can use data to drive the change process, but recognizing that data may be differentially viewed based on gender or lived experiences. And I give a reference down here that you might want to look at. So how do you get the data? You can document starting in your department, or you can use high-profile publications, which is what the work of Joya Misra did. And then you can also engage in changing the culture by recruitment and promotion and mitigating unconscious bias and setting specific goals and targets. So I'm going to skip this last word cloud and just sum up by saying that what I hope to share with you today is that it's important to define your strengths and bring your passion to your research and leadership roles, to build networks that include mentors, allies, and collaborators who'll support you to reach those goals, and that your career paths will, by definition, be filled with twists and turns. You will acquire knowledge and skills along the way that you can use. Always use these as your collective knowledge and skill set as a stepping stone, and embrace change and new opportunities, but also know your boundaries and limits. When you say yes to something, what are the conditions that you will do that job under? And then finally, please pay it forward and inspire that next generation. If you wish to contact me, my information is there, and I'm happy to share these slides with you. And I thank you for your attention and for coming today.
Thank you so much, Dr. Wandinger-Ness. We really appreciate your expertise and sharing. Does anybody have any questions, or if anyone needs to take off, please feel free to do so.
Yeah, people had to take off. We're
Yeah.
a little late getting started. I'm sorry if I took
no,
too
it's
long.
fine.
So.
And we will be sending out the evaluation we always send out, and appreciate anyone who could follow up.
Sure.
But,
Dolores, I'll share these slides with you. I don't know if you usually share them, but I'm happy to share them. There's nothing confidential. So if there's anything useful for people, I'm happy to share that. Okay.
Wonderful. Thank you very much. This was really, really lovely to see. I appreciate your time. Thank you.
Sure,
Thanks everybody for attending.
Bye. Have a wonderful holiday.
Thank you. You too. Bye.